120-year expiration date proved. Warning: science found a built countdown. Most humans won't beat 120, no matter what. In one lab dish, the cells just stopped. 46, 47, 48 divisions, then silence. No joke, the counter hit 0. Meanwhile, a naked mole rat shrugs off cancer, and a bowhead whale quietly pushes past 200 years. Plot twist: the same switch that protects you from tumors also slams the door on forever. Your body has guardrails, and trying to jump them tends to break things in weird ways. Imagine a world where cheating the clock is real, but the bill arrives as cancer. Rewind to where we first saw the counter tick. Roadmap and why this matters. Let's be real. Aging isn't just rust. It acts like a system with rules, and one of those rules draws a line near 120 years. We're gonna map how we found that line, what holds it in place, and what happens when you push it. Here's the road. First, the moment a dish full of normal human cells quit dividing, the Hayflick Limit. Then the fuse at the end of each chromosome, telomeres, and the enzyme telomerase that can rewind it for a price. Next, the zombie state called senescence that stops damaged cells but spreads trouble. We'll pull back to evolution's trade. Live fast, reproduce early, pay later. We'll meet outliers, naked mole rats, and bowhead whales, and see what they pay to bend the curve. We'll check the human ceiling with hard data from centenarians. And yes, we'll poke at the cheats. Telomerase therapy, partial reprogramming, and drugs that clear senescent cells. Some wins, some backfires. Why this matters: if there's a hard stop near 120, your best shot is more healthy years, not magic decades. And if you push past the guardrails, cancer waits. That's not a scare line, that's the trade. Open loops to keep in your pocket. One experiment that flipped medicine's view of aging. One gene tweak that looked like a miracle and then raised tumor flags. One simple habit cluster that, annoyingly, still beats the flashy stuff. Okay, back to the dish where the clock first spoke out loud. The dish that stopped dividing. Hayflick shock. Have you ever wondered when aging stopped being a foggy idea and started acting like a counter you could count? Picture this. 1960s. Leonard Hayflick at the Wistar Institute. Rows of flasks with normal human fetal fibroblasts. Nothing exotic, no special sauce, just careful counting. He tracks how many times those cells split. 30, 40, 45. Around 40 to 60 doublings, they slow, they flatten, they change their look, and then they refuse to divide. Not dead, not thriving, just done. People had assumed normal cells, given food and warmth, would go on forever like HeLa cancer cells. They didn't. Stakes, right there. If healthy cells have a built-in limit, aging isn't just damage piling up. There's a program that says stop. And once you see a program, you can ask what it's made of, who said it, and what happens if you push the buttons. Action as it unfolded. Hayflick keeps receipts. He freezes cells, thaws them years later, and shows their divisions add up across time. He shows this isn't contamination, not a bad batch, not a lab fluke. It's normal biology. Meanwhile, the field is still soaked in the belief that cells are immortal in culture if you treat them well. A belief fueled by Henrietta Lacks's HeLa line, which was actually a tumor, with its safeguards broken. That difference matters. HeLa is like a car with the brakes ripped out. It'll go far and fast, straight into a wall. Resistance comes quick. Senior voices push back. You must poison them. Your technique is off. Hayflick answers with data and a freezer full of proof. The WI38 cell line he helps establish becomes a workhorse for vaccines, including rubella. Quiet irony. The same cells that prove limits also help us cheat infectious deaths, letting more people make it to old age where those limits start to bite. Impact. The immortality vibe in cell biology takes a hit. Labs begin to separate normal from cancer in a serious way. The question shifts from how do we keep cells dividing forever to how do cells decide when to stop and what are they protecting? That pivot births a new language. Senescence, checkpoints, and soon, telomeres. Here's the twist that keeps the tension. Cancer cells blow past the limit. Most of them turn telomerase back on, the enzyme that rebuilds the ends of chromosomes. Others use an odd route called alternative lengthening of telomeres. Either way, they dodge the brakes. It works for them. For the host, it's disaster. So the limit looks less like a flaw and more like a safety fence. It keeps most of us from erupting into tumors when cells take too many risks. Zoom into a microstory. Picture Hayflick labeling a flask PDL-47, population doubling level 47, like a diary entry. He comes in after the weekend, checks confluence, adds medium, and waits. The cells look bloated, spread out, like they've settled into old age. They're alive, they're metabolizing, they even talk to their neighbors with secreted factors, but they won't cross the line into S phase. He marks the notebook. No further division. That one line becomes a pivot in how medicine thinks about aging, cancer, and what's even possible. Stakes ratchet again when you realize the human angle. If your tissues are built from cells that can only divide so many times, wound healing slows, organ maintenance gets thin, and the odds of breakdowns climb. But you also dodge a world where a hungry precancerous clone can sprint forever. The limit is a compromise. It's not your enemy, it's your bouncer. What nobody knew at the time was the exact counter. Why 40 to 60? Why not 10 or 100? To understand why cells keep score at all, we've got to look at the ends of your chromosomes. Tiny repeating sequences that act like the plastic caps on shoelaces. Every copy shaves them down. When they get short enough, alarms ring. That's where we're headed. Meanwhile, think about the cultural shift. Before this, aging was hand-waved as wear and tear. After this, people started asking, where's the clock? The answer didn't just live in chemistry, it lived in strategy. Your body's long war against rogue growth. The Hayflick Limit said the quiet part. To survive as a multicellular creature, you budget division, you ration youth, you retire cells before they turn. There's one more human beat to hold on to. The same WI38 cells that aged out of division helped make vaccines that saved millions of kids. Those kids grew up and met the lifespan ceiling more often. So the discovery that sets a limit also helps more people reach it. That paradox runs through this story like a seam. Every gain opens a new bottleneck. Every fix reveals the next weakest link. And here's where it gets crazy. If you can find the fuse, maybe you can splice it. If you splice it, maybe you add time. But as cancer showed us, splice too much and you light a different fire. So, to understand the counter Hayflick felt in his hands, we need to go to the tips of the chromosomes themselves. The fraying fuse that decides when the bomb doesn't go off. Telomeres, the fraying fuse at each chromosome tip. To understand the counter, Hayflick felt. Picture the end of every chromosome wearing a little cap. It's the same 6 letters repeated over and over. TTEG, stacked like beads. That cap is a telomere, and it's not there for decoration. It keeps the real genes from unraveling or sticking to each other. Now here's the catch. DNA polymerase, the copy machine, has a design flaw. It can't finish the last bit on the lagging strand. That end replication problem means each round of division shaves the tip. It's like cutting your hair with clippers that can't reach the last millimeter. You lose a little more every time. Stakes first, because they're real. Every time a skin cell repairs a nick, every time your gut lining renews, you spend a bit of telomere. When those repeats get short enough, the cell reads it as danger. Exposed chromosome ends look like broken DNA. Checkpoints flip. P53 and P21. Whisper. Stand down. The cell enters senescence or bows out with apoptosis. That's the fuse. Too short, and the system would rather stop than risk copying damaged code that could seed a tumor. It's protective, but it means tissues with high turnover run into maintenance limits with age. Wounds close slower. Organs recover less. That's the bill. Action as it unfolded in labs. Through the 1980s, Elizabeth Blackburn maps telomeres in a pond critter. Carol Greider finds telomerase, the enzyme that adds TTAG back on, and Jack Sostak helps show how these caps guard chromosome ends. Telomerase is a reverse transcriptase with its own RNA template, like a tiny factory that rebuilds the fuse. Most of your somatic cells keep it low or off. Germ cells, some stem cells, and plot twist. Most cancers flip it on. You can see the logic. Reproductive lines need immortality. Tumors steal it. Now, why should you care beyond textbook trivia? Because telomere length is one of the few cellular clocks you can actually read. In blood cells, shorter telomeres track with higher risk of heart disease, some cancers, and earlier mortality. Chronic stress, smoking, high inflammation. These don't just feel bad, they seem to tug telomeres down faster. It's not a one-to-one you lose 50 base pairs when you skip sleep kind of thing. And association isn't destiny, but as a signal of wear, it's useful. Here's a human beat. Imagine Grider on Christmas Day 1984, seeing a faint activity band on a gel, evidence of telomerase. That tiny smear would win a Nobel decades later and open a can of snakes. Because the moment you have a way to rebuild the fuse, you're tempted to rewind the clock. People tried it in mice. AAV viruses carrying TERT, the telomerase gene, into aged animals led to better heart function, improved muscle, sometimes longer median life. But even in mice, researchers watch like hawks for tumors, because that same enzyme is active in about 90% of human cancers. Rebuilding the fuse in the wrong cells isn't rejuvenation. It's fuel on a spark. Plot twist. The same liver that makes an old mouse's heart look fresher can hand a dormant pre-cancer a VIP pass. So you get hope with a hazard sign, not a fountain of youth. Complication time. Not all tumors use telomerase. Some use alternative lengthening of telomeres. Basically a recombination trick to copy and paste telomere sequences from one chromosome to another. It's messier, often seen in certain sarcomas and brain tumors. Either way, the path around the fuse is a cancer path. That's the trade written into the hardware. Meanwhile, lifestyle shows up as a quiet nudge on the shortening rate. People with lower systemic inflammation, steady glucose, good sleep, and strong social ties often show longer leukocyte telomeres for their age. Caregivers under heavy, chronic stress sometimes show shorter ones. Athletes aren't immortal, but higher cardiorespiratory fitness trends in the right direction. Again, signals, not guarantees. But if you're choosing levers, these matter more than a shiny supplement stack. Imagine swapping a cart full of powders for 8 hours of sleep and a barbell. Boring, yes. Viral for your biology, also yes. Impact on the big picture. Telomeres turned aging from a vague blur into a checkpoint with wiring diagrams. They gave us a handle to measure change, and a reason why the Hayflick Limit lands near 40 to 60 doublings. They also warned us that trying to brute force division by juicing telomerase everywhere lines up with cancer's favorite cheat. So our thinking shifts. If telomeres are the fuse, then senescence is what happens when the fuse burns low. A cell that won't die, won't divide, but won't shut up either. One last revealing detail to carry forward. Telomeres aren't the master clock of everything. Some tissues rely heavily on stem cells and keep telomerase humming in a tight, local way. Others, like neurons, aren't dividing much, yet still aged. So telomeres are a big dial, not the whole dashboard. That matters because our next stop is what short telomeres actually trigger. A state that can save your life from cancer in your 30s and make your joints ache in your 70s. Senescence, cells that don't die but won't work. So when telomeres run low or DNA takes a hard hit, the cell has a choice. Roll the dice and divide anyway, or pull the emergency brake. Senescence is that brake. Picture a cell that's alive, metabolizing, answering emails, but it's turned in its key card. It won't divide. It spreads out like it's flattening into the dish, ramps up stress responses, and starts blasting the neighborhood with messages. Helpful ones at first. Then noise. Stakes first because this touches real life. Senescent cells build up with age in fat, joint cartilage, blood vessels, even the brain's support cells. A few here and there are fine. Too many, and they clog the works. They secrete a cocktail called the SAFs, the senescence-associated secretory phenotype. That's a mouthful, but think of it as a leaky speaker system stuck on alert. Cytokines like IL-6 and IL-1, chemokines, growth factors, matrix-eating enzymes like MMPs. Nearby cells hear that, stress out, sometimes turn senescent too. Tissues inflame. Repair gets weird. You feel it as stiffness, slow healing, insulin resistance, frailty. It's not one villain, it's a choir that won't stop singing the same anxious chorus. Action, step by step. Short telomeres signal broken end to the damage sensors. ATM and ATR kinases light up. P53 and its sidekick P21. Put the brakes on the cell cycle. The P16 Inc4A RBAL pathway locks the door. Mitochondria cough out a bit more reactive oxygen. That feeds back into the stress loop. Chromatin, the way DNA is packed, rearranges, turning on the SASP playlist. The cell starts pumping out IL-6, IL-8, TGF-SPU, GM-CSF, MMP-1, MMP-3. Meanwhile, it resists death by upshifting survival proteins like BCL2 and BCL-XL. So now you've got a tough, loud, retired cell that won't leave the building. Here's a human scale moment. A mouse that's been creeping across the lab floor like it's 80 gets an intervention. It's part of the Inc attack model. Basically, scientists gave mice a genetic self-destructive senescence switch they can flip with a drug. They clear a chunk of these zombie cells. Within weeks, the mouse moves cleaner. Its treadmill time ticks up. Its heart and fat tissue look younger under the scope. Later, researchers tried drug combos like dasatinib plus quercetin. One's a cancer drug, one's a plant flavonoid, to hit senescent cell survival wiring. Old mice jump higher. Their fur looks fuller. Median lifespan nudges up. It's not magic, it's housekeeping. Impact shows up in people too, early and cautious. Small pilots report osteoarthritis pain dipping after. Senolytic cycles. Some patients with pulmonary fibrosis breathe a bit easier. Frail older adults post better walking speed. Diabetic kidney disease shows hints of improvement. None of this is a cure, and the trials are still sorting dose, timing, and who benefits. But it suggests that clearing a noisy minority can let the majority of cells work better. It's like pulling cars off a jammed lane. Traffic flows, even if the road didn't get wider. But, and this but matters, senescence isn't a mistake evolution forgot to delete. It's a feature with a price tag. Those same P16's P21 breaks stop tumors in their tracks. A wound needs a burst of SAPs to recruit immune cells and kick off repair, and then it needs that burst to fade. In embryos, a senescence-like program helps shape tissues. So if you carpet bomb senescent cells, you might blunt cancer risk in one place, but trip it in another. You might fix a knee and mess up how a cut closes. Timing and specificity are everything. Meanwhile, the immune system is supposed to patrol for these cells. With age, that patrol gets sloppy. Immunosenescence on top of cellular senescence. Fewer natural killer cells show up. Macrophages chill when they should clean. So senescent cells linger longer, seeding more SAPs, feeding chronic inflammation, the inflammaging you hear about. It's a loop. Break it at the right spot, you help. Break it at the wrong one, you invite trouble. Get this? Cancer again sits on the other side of the teeter-totter. If you loosen the brakes everywhere to feel younger, precancerous clones notice. If you tighten them hard, tissues that need turnover, gut, skin, blood, start to wear thin. The body splits the difference. Accept some senescence, keep a lid on clones, live long enough to raise kids and maybe grandkids. After that, the math gets fuzzy for evolution. That's why these trade-offs stick. So senescence is the price of safe living in a multicellular crowd. It protects you from a runaway few by retiring the many a little early. And if you can clear the worst offenders, carefully, you can buy better function without yanking out the fire alarms. The question is why nature picked that bargain in the first place, and what else it gave up to keep us safe long enough to pass the baton. The evolution bet. Die so your kids don't. Here's the part that sounds harsh but explains almost everything. Evolution doesn't care how you feel at 80. It cares whether you left kids who can leave kids. So it spends big on what helps you survive and reproduce early, and it cuts corners on what happens after. That's the quiet bet under every limit we've seen. Telomeres, senescence, the brakes that hold back cancer but also slow repair. Let's dive into antagonistic pleiotropy, which is a fancy way to say one gene wears two faces. Early on, it's your hype man. Later, it's a tax bill. Think growth and sex hormones. They push muscle, bone, fertility, and fast, wound healing when you need to compete, mate, and raise babies. Same signals also nudge cell division and metabolism in ways that, decades later, hike your cancer risk and strain your heart. From evolution's seat, that's fine. You already passed the baton. The late downside is invisible to selection because selection fades with age. Past your peak reproductive years, the spotlight moves on. Now add the disposable soma theory. Thomas Kirkwood's "you are a budget" idea. Your body is juggling calories and micronutrients and time between three buckets: grow, reproduce, maintain. Maintenance is boring, but pricey. DNA repair, protein quality control, immune policing, all the quiet stuff that keeps cells tidy. In safe environments with low external danger, it's worth investing more in upkeep. You might live to use it. In risky environments, predators, infections, accidents, it's smarter, genetically speaking, to pour fuel into early reproduction. Why fix the roof if the house might burn down this season? So lineages evolve a maintenance spend that fits their world. We got enough to clear childhood, hit puberty, raise offspring, maybe help with grandkids. Not enough to keep everything pristine into year 150. You can see these trade-offs in nature, like signposts. Pacific salmon are the brutal version. Sprint upstream, spawn, crash. Their bodies go all in on reproduction, catabolizing muscle and organs to feed that final push. Not subtle, but honest. Maximum reproduction, zero maintenance. Opossums on predator-heavy islands age faster and die younger than their mainland cousins, living with fewer threats. When the outside world kills you early, there's no payoff for high repair budgets. Bats flip the script. They're small, but they fly, which makes them safer from predators. With lower external depth, selection sees late life more clearly. Over time, bats evolve better maintenance, tighter inflammation control, unusual immune tolerance, and live decades. Same with birds. Lower extrinsic mortality buys time, so the genome puts money into the maintenance line item. Meanwhile, big, long-lived mammals like elephants and whales showcase how expensive that shift is. You don't get to live 2 centuries with a giant body unless you solve cancer several ways at once. Bowhead whales carry tweaks in DNA repair networks and cold-responsive proteins that keep genomes cleaner under stress. Elephants stock extra copies of p53, like genes to kill sketchy cells on site. These aren't upgrades you slap on in one mutation. They're whole system rewrites paid for by slow reproduction, long childhoods, and careful ecologies. The price is years between babies and a huge parental investment. The reward is time. Back to us. Humans look like we hacked evolution because we outlived most hazards with culture. Fire, clothes, medicine, social care. But biology can't rewrite itself on the fly just because we invented seatbelts. Our wiring still reflects a world where infections killed kids and a broken ankle could end a life. So our maintenance budget sits where it sat. Good enough to reach and support reproduction, not bulletproof at 110. Telomeres shorten, senescence climbs, mitochondria fray. The clocks tick because they're part of the safety plan that worked when cancer was a bigger short-term threat than arthritis. Here's where it gets tricky. Some late life traits that look bad might be echoes of early life wins, not mistakes. High insulin signaling helps you grow. Dial it high for decades and you get insulin resistance. Strong inflammatory responses clear pathogens when you're young. Lean on that same setting in your 70s and you get chronic inflammation that cooks tissues slowly. Even bone. Dense bones help a young athlete. By later life, that same biology can calcify arteries. The dial positions are compromises. They weren't tuned for modern lifespan with antibiotics and statins and colonoscopies, so why a ceiling around 115 to 120? Not one lever, many. Antagonistic pleiotropy gives us late life liabilities bundled with early life assets. Disposable soma caps the maintenance spend. Then multicellular rules add more constraints. You need strong cancer policing, which means rationing division. You need flexible immunity, which risks autoinflammation. You need regeneration, which risks miscopying. Push any one dial hard, say telomerase everywhere, and the others scream. The system stays near a safe operating window because drifting far outside, it makes you either cancer-prone or too brittle to heal. Imagine a car designed to finish a tough rally, not circle the track forever. The suspension is strong, but not serviceable mid-race. The engine's tuned for torque, not infinite hours. You can nurse it past the finish with mechanics and luck, but the blueprint wasn't drawn for lap 500. That's us. Culture and medicine are our pit crew. We keep the car running longer than the bare spec, but the parts were chosen for a different race. Could selection move the line if we lived many millennia in cushy safety? Maybe. Slowly. But evolution works through reproduction, not personal desire. And as soon as you try to engineer around these bounds in one generation, flip telomerase on, kill all senescent cells, you collide with the reasons those bounds exist. The bet nature made wasn't die young, it was guard the young, then accept wear. We're the first species to argue with that bet at scale. And every argument runs into the same fine print. To buy forever, you must out-engineer cancer, tame inflammation, and fund maintenance in every tissue without breaking identity.
And oddballs, species that bend the curve. While our dials sit near 120, Nature's Workshop has a few prototypes that mess with the settings. The point isn't to copy-paste them into humans. It's to see what they paid to bend the curve, because every cheat comes with a tab. Start underground with the naked mole rat. It looks like a pink peanut with teeth, but its biology is loaded. In lab after lab, these rodents show near negligible rises in mortality for much of their long mouse-beating lives. They rarely get cancer. Plot twist. One reason is a goo. Their tissues are packed with high molecular weight, hyaluronan, a supersized sugar polymer. That stuff turns on contact inhibition early. When cells bump neighbors, they stop dividing sooner and tidy up. Precancerous clones can't elbow their way to a mass. It's like adding extra fire marshals to every hallway. Action you can picture. Researchers grow naked mole rat fibroblasts next to mouse or human cells. The mole rat cells spread, then halt at low density. Try to transform them with oncogenes that push normal cells over the edge, and they shrug. Yank out or digest that giant hyaluronan, and suddenly the cells act more like ours. They'll misbehave under the same provocation. The message is blunt. Their brake pads are thicker. Great for cancer resistance, which buys time. But impact isn't all upside. Naked mole rats grow slow, reproduce late for a rodent, and live in eusocial colonies where only a queen breeds. Most animals in that tunnel network never pass on genes. Stability, low injury risk, and tight social order let them afford that strong anti-cancer setting. Their cheat is really a lifestyle and ecology package. Low extrinsic danger, low metabolism pace, high maintenance on key safety pathways. Translate that to humans? We don't have their tunnels, their hierarchies, or their exact extracellular matrix tricks. We can learn principles. Early contact inhibition and clean extracellular environments help, but ports aren't plug and play. Now go cold and massive. Bowhead whales. 200 years in Arctic waters, with a body the size of a building, and yet not drowning in cancer. That spits in the face of simple, more cells, more cancer logic, which is why people talk about Peto's paradox. How do they cope? Evidence points to tougher genome maintenance. Their cells show enhanced DNA repair capacity and a stress response wired for slow, steady fixes in the cold. One standout is a cold-inducible RNA binding protein, CIOPP, that seems tuned differently. In bowhead cells, CIOP ramps up efficient repair signaling and keeps transcription tidy when things get chilly and stressful. That reduces the noisy mistakes that, in us, pile up into mutations. Action in the dish. Take bowhead fibroblasts, stress them, radiation, chemical hits, and compare to human or mouse cells. Bowhead cells activate repair pathways fast and clean. Assays for double-strand break repair light up. Fewer lingering foci hang around. Meanwhile, telomerase activity in bowhead somatic cells isn't wildly high. The point isn't endless division. It's precision repair, apoptosis when needed, and anti-inflammatory restraint. Stack that with other tweaks. Altered insulin, IGF signaling, lipid handling for cold seas, and you get a body that avoids both runaway clones and chronic simmering damage. Impact for the big picture. Whales pay with the slowest life history. Late sexual maturity, long intervals between calves, huge parental costs, and a safe niche with few predators. That safety lets selection see late life and reward maintenance. It's disposable soma turned inside out. When outside risk is low for centuries, maintenance gets the blank check. The trade is growth and reproduction speed. You can't have both. Slide to another oddball. Elephants. Different path, same goal. They carry extra copies of tumor suppressor genes like p53 family members. When DNA looks sketchy, elephant cells don't hesitate. They self-destruct more readily. That's a pro-apoptosis setting baked into the spec. Great for stopping cancer in a huge body. The cost? Likely slower regenerative responses and a need for careful tissue architecture so you don't overprune. Micro moment for context. Hydra. tiny freshwater animals that seem to show negligible senescence in the lab. Stem cells churn, bodies remodel. No obvious age-related slide. Sounds like immortality until you zoom out. In the wild, hydra die from predators, infections, and winters. Their no-aging story is buffered by ecological death. If you remove outside risk, their internal maintenance looks timeless. Put them back in a pond, and nature ends the run for other reasons. Lesson: biology can sustain, but environment decides if you ever cash that check. Curiosity gap: if a creature can dodge aging, why doesn't it rule the world? Because everything else tries to eat it first.
So what do all these cheats share? They buy time by shoring up failure points that kill us.
Cancer, chronic inflammation, sloppy repair. They do it with concrete mechanisms. High molecular weight hyaluronan that enforces early stop signs. Upgraded DNA repair orchestras tuned by proteins like CIRBP. Duplicate tumor suppressor strategies. And every upgrade sits inside a life history that slows the whole system. Fewer babies, later babies, safer habitats, slower growth. No joke, longevity isn't one gene, it's a budget and a neighborhood. Here's the human takeaway without fairy dust. We can't turn into whales, but we can copy principles. Build therapies that mimic clean repair without feeding tumors. Enforce early contact inhibition in tissues that get sketchy. Cool chronic inflammation without smothering immunity.
And accept the trade. Any real move to push past our ceiling has to solve cancer first, then the quiet background errors. The outliers don't break the rules. They pay to rewrite them. Everything's about to feel personal again, because our species already flirts with a soft ceiling. Are we inching it up or bouncing off it with better pit stops? The human ceiling, data from centenarians and records. While the whales and mole rats show what's possible, our species keeps bumping a softer ceiling. So are we hitting a wall, or did we just not build the right ladder yet? Let's open the ledger where the numbers live. Death records, birth cohorts, and the rare humans who make it past 110. Stakes first. This isn't trivia about the world's oldest grandma. It's the question of whether medicine is adding years to the end or just packing more health into the years we already had. If there's a practical ceiling near 115 to 120, you plan your life one way. If it's drifting upward, you plan another. Action in the data looks like this. Plot human deaths by age on a log scale, and you get a clean slope through adulthood. Mortality doubles roughly every 8 years. That's the classic Gompertz curve. But keep following the line into the 90s and you notice a bend. The slope softens. Past about 100, death risk still climbs, but it doesn't rocket like before. People call it a late-life mortality deceleration, or a plateau. Sounds hopeful, right? Here's the catch. The absolute annual risk is already huge. At 110, your odds of dying this year can hover around 50%. A coin flip every birthday. The slope calming doesn't mean safety. It means you're already on the cliff edge, and the cliff stays the same height for a few more steps. Now zoom into supercentenarians, 110 and up. A small club, and for good reason. If you take countries with good vital records and clean out fraud and mistakes, you still get only a few dozen living at any time. The validation work matters more than it sounds. Typos, identity swaps, and reused documents make fake immortals. When researchers tighten validation, contact families, check birth certificates, cross-match censuses, the numbers thin, but patterns stabilize. Most people who make it past 110 die before 115. A handful nudge 117, 118. Gene Kalman's famous 122 stands alone like a radio tower on a flat plain. Let's talk Kalman, because her record is both a beacon and a lightning rod. She lived in Arles, France, with a well-documented life story. Met Van Gogh, sold him pencils, smoked into her hundreds, joked with reporters. Her papers looked solid, and longevity researchers used her as a proof that 120 plus was at least possible. Years later, a small group questioned the record, suggesting a potential identity swap with her daughter to dodge taxes. It made headlines, then got batted around by demographers. The consensus in the field still leans toward record holds, because the documentation chain and eyewitnesses line up, though it's fair to say, the debate sharpened how carefully we vet extreme ages. Either way, the broader point stands.
If a modern, well-fed, medically supported society produced only 122 in all recorded history, that's not a trend. That's an outlier. Impact in the tail shows up in statistics built for extremes. Use extreme value theory on the oldest ages, and you can estimate a moving frontier, what the maximum looks like for a given birth cohort. Across countries that have kept people alive better and better, the frontier inches, but it doesn't leap. Life expectancy at birth soared in the 20th century because we crushed infant deaths and infections. Life expectancy at 60 rose because we beat heart attacks with blood pressure, drugs, and statins, and we caught cancers earlier. But the maximum verified ages didn't march up in step. You can picture it as a wave that got smoother and taller in the middle, while the crest at the very top barely shifted. Here's a human beat to make that real. Meet a woman turning 105 in Okinawa. She eats with her friends, gardens a little, naps in the sun. Her blood pressure is fine. She'll probably see 107. She's part of a cluster where many reach 95, many hit 100. The community adds years of good function, not just time. But even there, 115 is rare, 120 basically unseen. The ceiling holds, even when the culture does almost everything right. Complication, because we need honesty. Medical progress hasn't stopped helping at older ages. We've got better pacemakers, joint replacements, cancer immunotherapies, we've compressed morbidity, people spend fewer years in disability. That's huge. It's just not the same as shifting the maximum lifespan line. Pushing that would mean consistently stacking new 120s, not one every other century. Meanwhile, the biology we walk through makes sense of the chart. Telomeres, senescence, immune drift, mitochondrial fray, epigenetic clocks. They don't all collapse at once, but past 110, you're juggling dynamite. Your stem cell reserves are thin. Your repair budgets are low. The plateau shows physics, not mercy. So are we stuck? Not quite stuck. Channeled. The path up requires taming cancer while maintaining regeneration, cooling chronic inflammation without erasing immune defense, and resetting cellular software without wiping identity. Until we do that in many tissues at once, the human ceiling sits near 115 to 120. The smart money today is on making more people hit 95 feeling 75, not promising 140. And honestly, that's still a win. Cheating the clock, telomerase, CRISPR, and tumor trouble. So we've got a fuse, we've got brakes, and we've got a ceiling. What if you just turn the fuse back up? That's the telomerase temptation. Researchers tried it the careful way. Old mice, adeno-associated virus vectors carrying TERT, the gene for telomerase. Dose the animals late in life so you're not juicing precancerous clones for decades. And get this, the hearts pump better. Cardiac muscle looked younger under the microscope. Some studies showed improved metabolic profiles and small bumps in median lifespan. For a minute, it felt like we'd found a lever. But the lab is not a wish factory. Tumor monitors ran non-stop because telomerase is cancer's favorite tool. Around 90% of human tumors flip telomerase back on to dodge the Hayflick fence. So every look the heart's happier came with, and we're scanning for sneaky clones weekly. In mice with tight dosing windows, risk didn't always explode. In a human with decades of field time and millions of tiny lesions across tissues, you don't get that kind of slack. The stakes are clear. Turn telomerase on. Tissues regain capacity to divide. Therefore, any hidden precancer can sprint. Keep it off and tissues stall. There's no free lane. You need a map, not a gas pedal. What if you don't just fuel the fuse? What if you rewind the software? Enter partial reprogramming, the wild idea behind Yamanaka Factors. October 4th, SOX2, KLF4, C-MyC, OSAC-M for short. Turn them on fully and you blast cells back to pluripotency. They forget who they are. That's bad inside a living organ. But what if you pulse them, low and brief, like tapping refresh without nuking identity? In progeroid mice, the ones that age like they're fast-forwarding, cyclic OSCAT-M expression, improved tissue repair, smoothed epigenetic marks, and extended lifespan. In normal mice, careful cycles show better muscle regeneration and faster wound closure, without obvious cancers, if the dose was right. Plot twist: if the dose wasn't right, identity blurred. teratomas formed, and everything you fear about stem cell chaos showed up. Timing, tissue, and threshold. Those three decide if you bought youth or lit a fuse. Right now, the field is inching into first-in-human territory, but with guardrails. 2024 and 2025 have seen early trials and preclinical packages aiming at single tissues, eye, nerve, muscle, where local delivery and tight control lower risk. Think transient OASK, dropping sync to cool tumor drive. Put under an inducible switch that only flips in that tissue, and only when the drug key is present. It's not bathe the body, it's patch this room. And even there, the monitoring stack is heavy. Imaging, circulating tumor DNA, biopsy windows, and off switches baked in so you can kill the program if cells go weird. No joke, the consent forms read like pilot checklists, because they should. CRISPR sits beside all this like a scalpel we're still learning to hold. Could it tweak senescence wiring so cells land softly instead of shouting saps? Could it harden tumor suppressor pathways without breaking regeneration? People are trying edits to P16's P21 regulators or to DNA repair genes that sag with age, aiming to tune responses rather than slam them. There are also CRISPR-based epigenetic editors, no cuts, just marks, that try to dial clocks back a tick or two. In mice, you see hints. Cleaner repair in certain tissues, smaller scar sizes after injury, a cooler inflammatory tone. But every therefore has a but. Edit a tumor suppressor too strong and your gut stem cells phone it in. Healing stalls. Ease the brakes too much and a pre-malignant patch in the esophagus gets bold. The system bites if you don't respect its trade-offs. Plot twist. The genes you want to anti-age are the same ones your body uses as anti-cancer tripwires. Micro story from a lab bench. A team pulses OSK in old mice with a viral vector designed to hit muscle. For 2 weeks, nothing dramatic. Week 3, treadmill tests improve. Muscle fibers look less ragged. Mitochondrial stains brighten. They celebrate. Then a mouse in a different cohort grows a lump under the skin. Pathology shows a mixed tumor near an injection site. Was it the vector, the dose, the mouse? They halt, retool the promoter, drop CMIC, add a suicide gene that triggers cell death if markers of uncontrolled proliferation spike. Run again, slower, fewer gains, fewer scares. This is how the sausage gets made. Every bump forward is tied to a reminder. Rejuvenation and cancer use overlapping roads. Telomeres, reprogramming, CRISPR. None of them are magic. They're pattern edits. And because aging isn't one lever, even a good edit ripples. You lengthen telomeres in hematopoietic stem cells. Therefore, more blood cell divisions are possible. Therefore, infection recovery might improve. Therefore, a stressed clone in the marrow could also find extra runway. You pulse OSCA in the eye for optic nerve disease. Therefore, some glial cells reset a bit. Therefore, scar programs loosen. Therefore, a latent herpes infection might wake up. It's a web. If you don't plan for the web plans for you. So what's the shape of a sane cheat? Local, transient, monitored, and stacked with fail-safes. AAV TERT in an old heart, not a teenager's whole body. OASK without CMIC under a tissue-specific, drug-inducible promoter with a kill switch. CRISPR that nudges a repair pathway up 10%, not a double knockout that makes you a science headline. And even then, you pair it with the boring stuff, exercise, sleep, vaccines, because lowering background inflammation and mutation pressure makes every fancy trick safer. Honestly, the story here isn't don't try, it's know the cost. Biology gave us guardrails for a reason. If you plan to hop them, you better bring armor, a map, and a way back. Senolytics, taking out the cellular trash. We've tried fueling the fuse and nudging the software. What if the simpler move is cleanup? Kick out the loud retirees messing up the neighborhood and let the rest of the block breathe. That's the Senolytics idea. Drugs that spot senescent cells by their survival wiring and push them over the edge. Stakes first. If you can lower that background noise, the SAS, without wrecking repair, whole tissues might work better. But those same cells help heal wounds and police early tumors. Clear too much, wrong time, wrong place, and you pay with slow healing or missed threats. It's a tightrope over a fireworks factory. Action as it started in mice was almost cinematic. In the ink attack model, scientists stitched a self-destruct handle into cells that turned on P16 ink 4A, a senescence marker. Give the mouse a drug, the marked cells die. Old animals moved smoother, their hearts and kidneys scored better, cataracts delayed. Proof of concept: fewer zombies, cleaner function. Then came off-the-shelf hits. Researchers noticed senescent cells lean on anti-apoptosis proteins, BCL2, BCLXL, like crutches. So they tested combos to kick the crutches. Dasatinib, a cancer drug that targets tyrosine kinases, plus quercetin, a plant flavonoid with a grab bag of targets, worked together on different senescent cell types. In aged mice, a few intermittent doses trimmed senescent burden, eased inflammation, and popped median lifespan up a notch. Fisetin, another flavonoid, showed promise too, with cleaner safety signals in rodents. Zoom into a lab microstory. A cage of creaky whisker-gray mice gets 2 days of dasatinib plus quercetin, then nothing for weeks. The change isn't Benjamin Button, it's subtler. Grip strength ticks up, treadmill time extends. Under the microscope, fat tissue secretes fewer inflammatory signals. Bone shows better remodeling. Heart fibrosis softens. The team runs cytokine panels. ILA6 and MMPs slide down. They smile, then they run tumor screens, glucose tests, and wound healing assays. Because any gain has to clear those hurdles. In this round, the balance looks good. Impact creeps into human trials, small and careful. In idiopathic pulmonary fibrosis, a brutal lung disease tied to scarring and age, short courses of dasatinib plus quercetin nudged physical function. 6-minute walk distance, sit-to-stand tests in the right direction. Early osteoarthritis pilots hint at pain dips after senolytic cycles. In diabetic kidney disease, biomarkers of function shift a little. Frail older adults post faster gait speeds after intermittent dosing. These are signals, not slam dunks, but they point one way. Clear, a subset of senescent cells, and some people feel and function better. Meanwhile, drug hunters sharpen the tools. If senescent cells prop themselves up with BCL2, BCLXL, then BH3. Mimetics, drugs that pry those proteins off, should be potent. Navitoclax hits that axis and does hammer senescent cells in preclinical work, but platelets also rely on BCLXL, so bleeding risk shows up. That's a trade doctors actually see. So teams try to split hairs, targeted versions, lower pulses, or newer molecules that dodge platelets. Others go the sniper route, antibody, drug conjugates that lock onto surface markers enriched on senescent cells, deliver a toxin right there, then clear. If you can keep the blast radius tiny, you get the trash out without wrecking the carpet. Complication you can't skip. Senescence isn't one thing. A senescent fat cell doesn't look like a senescent endothelial cell. Their SAS mixes differ, their survival wiring differs, their jobs differ by context. In wound healing, a timed burst of senescent signals recruits immune cells and growth factors, then fades. Hit that window with a senolytic, and you might slow closure. In embryonic development, senescence-like programs sculpt tissues. You probably don't want to carpet bomb that biology in a pregnant person. The immune system also uses senescence toggles to throttle responses. If you flatten them, infections might get freer lanes. Another wrinkle. How do you know you actually hit the target? You can't biopsy everyone's knee and heart every month. So trials stack proxies, circulating SASP factors drifting down, imaging inflammation cooling, functional tests improving. Some groups add PET tracers that light up senescent-rich regions, giving before and after snapshots. Others track single-cell RNA signatures in blood to see if senescence-leaning immune cells thin out. It's a lot of instrumentation for a simple story. Did we take out the trash? Here's a human-scale beat. A retired teacher with stiff knees joins a study. 2 days on, weeks off for 3 cycles. She doesn't wake up 20 again. She does notice stairs feel less like a negotiation. Her morning stiffness shortens. She calls her daughter and says, "It's like someone turned the volume down." That's the sap fading. The clinic draws blood, numbers slide in the right directions. The team high fives, then schedules skin punch checks to make sure wounds still close on time. They do. A hair slower on week 1, normal by week 3. The window matters. Where this heads in 2024 to 2025 looks like specificity and scheduling. Dose low, pulse, and pick indications where senescence is more villain than hero. Fibrotic lungs, diabetic kidneys, chemo-age bone marrow, radiation-scarred tissue. Pair senolytics with xenomorphics, drugs that don't kill senescent cells but quiet their SASP. so you get silence without as much collateral. Think repologs, JAK inhibitors, or even lifestyle levers that cool inflammation. And always screen for cancer risk, because a world with fewer senescent sentinels could give precancerous clones a little more air. Honestly, Senolytics feel like good housekeeping, not a time machine. They won't push 120 to 140 alone, but by lowering the noise, they make every other system work closer to spec. Mitochondria hum a bit better, immune patrols move with less static tissues, heal in cleaner arcs. In a body built on compromises, sometimes the smartest move isn't a new engine. It's clearing the filters
Systems that age you,
so the one you've got can
quietly,
still breathe.
Mitochondria, proteins, immune drift. While we're arguing with telomeres and zombie cells, a quieter crowd is shifting the floor under our feet. Think of your body like a soundboard in a studio. Aging isn't one switch. It's a dozen sliders moving a little each year. Mitochondria leaking, proteins misfolding, immune cells drifting, epigenetic labels smudging. None of these screams by itself, but together they change the song. Start with mitochondria, the little power plants. When you're young, they're crisp. Tight membranes, clean electron flow, ATP on demand. Over decades, the inner membrane gets dinged, complexes lose efficiency, and electrons leak like water from a cracked hose. Those leaks mean reactive oxygen species, tiny sparks peppering the area. Not a firestorm, more like static that messes with proteins and lipids. Mitochondrial DNA, tucked close to the action and light on repair tools, picks up mutations. The network compensates. Fuses, fissions, swaps good parts, but the average unit runs sloppier. Muscle tires faster, neurons struggle with long-haul signaling, and your get up and go needs a coffee and a pep talk. Now proteins. Your cells fold them precisely, then watch them like hawks. Chaperones shepherd new chains. Proteasomes chew up junk. Autophagy recycles broken gear. With age, this proteostasis system loses its edge. Chaperones show up late. Proteasomes drag. Autophagy stalls like a truck on a hill. Misfolded proteins clump. Amyloid here, tau tangles there, lipofuscin granules gumming up the works. It's not just brains and plaques. Everywhere, the background clutter rises. Enzymes run slower, receptors miscue. The cell's to-do list gets buried under spam. Meanwhile, the immune system, our beloved bouncer, changes the guest list. The thymus, the training camp for T cells, shrinks after puberty like a deflating balloon. Fresh recruits drop. The blood fills with veteran T cells that remember old viruses a little too fondly, and new ones not at all. B cells skew toward weird clones. Natural killer cells hesitate. You get two things at once. Slower responses to new threats, and a low-grade hum of inflammation, because some of those old cells won't shut up. That inflammaging feeds senescence and proteostasis stress, which in turn feeds more inflammation. Nice little loop. Here's where the story braids. Mitochondrial leaks goose the SASP in nearby senescent cells. Misfolded proteins jam autophagy, which was also supposed to keep mitochondria tidy. The immune patrol that should clean senescent cells is tired, so more senescent cells linger, secreting MMPs that chew extracellular matrix, making tissues sloppier. Skin sags, vessels stiffen. Each slider nudges the next. Microbeat from a classic experiment that blew minds, because it felt like sci-fi. Parabiosis. 2 mice, one young, one old, surgically share a blood supply. The old mouse heals cuts faster. Its muscle stem cells wake up, the heart looks a little friskier. The young mouse? Some functions dim. For a hot minute, people shouted, "Young blood cures aging." Then the plot thickened. It wasn't a magic sauce so much as a dilution game. The young circulation diluted pro-aging factors in the old, TGF beta family signals, inflammatory chemokines, while adding some supportive cues. The young mouse now swam in the old's noise. Some of its stem cells heard, "Stand down." The lesson wasn't vampire lore, it was network dynamics. Change the mix and the sliders move together. Zoom in on epigenetics without stealing the next act's thunder. Your DNA sequence stays, but the labels, methyl groups, histone marks drift. Stress, inflammation, mitochondrial status, even proteostasis problems whisper into the nucleus and nudge those labels. Some genes you need in late life get muffled. Some you prefer quiet perk up. The clocks built from these marks track that drift frighteningly well. They're not just calendars, they're thermometers for the system's simmer. Curiosity gap.
If the labels move, can we move them back without breaking the machine? Impact on daily life is boring and brutal. A 70-year-old catches the flu and takes a month to feel normal. A 75-year-old lifts a suitcase and tweaks a back that would have shrugged it off at 30. A small cut turns into a slow-healing sore because local stem cells are listening through static and the immune follow-up brings too much noise. Not calamity, cumulative friction. Here's where the temptation is to fix one slider hard. Supercharge mitochondria with a supplement stack, drown inflammation with a blanket drug, ramp autophagy with fasting and rapologues. Some of that helps. Exercise, for instance, is a mitochondria whisperer. It pushes biogenesis, prunes the worst units, and tightens the whole network. Sleep clears protein waste from the brain's lymphatic system. Vaccines tune immune drift by preloading blueprints for threats you're slow to meet. These are small, system-aware nudges, but when labs try a heavy hand, the network complains. Amp up mitochondrial turnover too hard and some tissues starve while they remodel. Oversuppress the immune system, and you swap inflammaging for infections that take you out faster. Smash proteostasis with blunt antioxidants, and you might blunt the signals that tell your body to adapt to exercise. It's that same theme. Distributed control beats, single knob fantasies. Human moment to land it. A granddad starts light resistance training, swaps late night doom scrolling for sleep, gets his vaccines on time, and adds a high fiber breakfast. 6 months later, he climbs stairs without the handrail. Labs show lower CRP, better A1C. He didn't rewire evolution. He slid half a dozen sliders by 10% each. That's the kind of math that moves how you feel, how you heal, and how many good years you stack. So yes, telomeres and senescence set guardrails, but the quiet systems decide how bumpy the road feels inside those rails. And speaking of quiet clocks, there's a deeper counter woven through all of this, the software itself drifting and sometimes resetting. The deeper clock, epigenetic drift, and biological age. So the DNA letters mostly stay the same. What changes is the labels stuck to them, the software. Imagine your genome as a giant switchboard with post-it notes telling which switches to flip. Over time, some post-its peel, others stick where they don't belong. That's epigenetics. Chemical tags like DNA methylation and histone marks that tell genes when to speak up or shut up. The tags drift. The song changes. Here's the twist that made biologists sit up. You can measure that drift like a clock. Not a vague, hand-wavy, you look tired clock. An actual statistical model that reads methyl groups at specific spots and tells you a person's biological age. Steve Horvath pulled the first big one together using data from many tissues. Feed in methylation at a few hundred CpG sites, and out pops an age that's often closer to how someone functions than the candles on their cake. A 70-year-old who trains, sleeps, and keeps inflammation low might clock in at, say, 63. A stressed 45-year-old caring for a parent with dementia might read older than their driver's license. Caregivers have shown this in studies. Chronic stress nudges the needle. Why does that matter? Because clocks built on methylation can predict risk. People older than their calendar age by the clock tend to face higher odds of heart disease, certain cancers, frailty, and earlier death. Newer versions don't just estimate age, they estimate pace, how fast you're aging right now per year. That's wild. It's also useful because it gives a way to test whether a change, sleep, exercise, a drug, nudges the slope. Action in labs gave us the freakiest part. Full reprogramming with Yamanaka factors wipes the slate. A skin cell becomes a stem cell, and its methylation clocks crash back towards zero. That's not what you want in a person. Cells would forget their jobs. But partial reprogramming, short pulses without going all the way, nudges clocks backward while keeping identity. In mice, OSK cycles smoothed methyl marks linked to aging and restored function in muscle and nerve. Even outside living animals, you can take old human cells in a dish, run a careful reprogramming pulse, and see epigenetic age drop while the cells still act like fibroblasts. It's like defragmenting a hard drive without deleting the operating system, if you do it right. Meanwhile, everyday life keeps writing to the software. Inflammation presses certain methylation sites. Mitochondrial status whispers to histones. High glucose streaks marks across promoters that handle stress responses. Exercise, on the flip side, scribbles helpful edits. It opens chromatin around genes for mitochondrial biogenesis and antioxidant defenses, then closes junk that pushes the SAS. Sleep does cleanup runs that help reset some gene programs, especially in the brain. Sunlight, insane doses, scribbles vitamin D, linked notes that touch immunity. Social connection, no joke, tracks with better biological age metrics, likely through lower stress hormones and cooler inflammatory tone. The software listens to the whole orchestra. Impact, right now, looks like 2 lanes. Lane 1 is measurement. Use clocks to flag people aging faster. Then offer levers that slow the pace. Muscle, sleep, vaccines, nutrition, glucose control, less smoke, less stress. Not sexy, but the clocks move. Lane 2 is intervention. Can we edit the software safely? Labs are tinkering with CRISPR-based epigenetic editors that don't cut DNA, but add or erase methyl groups at targeted loci. In a mouse liver, you can dial a stress gene down a notch without breaking the code itself. It's early, but it hints at programming aging dials, not just reading them. Here's a micro story. A study enrolls middle-aged adults, mixes strength training, brisk walking, more fiber, earlier dinners, and coaching to cut. Nightly rumination. 12 months later, blood hits the sequencer. The Horvath-style clock says their biological age barely budged, even dipped the hair, while match controls drifted 2 years. The team doesn't high-five like they cured death, but they nod. Pace matters. A slower slope today pays off a decade from now. Complication, because this isn't magic. Clocks can be fooled. A cancer can hijack methylation and look young in one readout while being very much a problem. Weight loss can shift clocks quickly, but if you did it by crash diet, you might have traded muscle for points on a graph. Different clocks measure different things. Some track straight age, others track mortality risk, others track pace. People love single numbers. Biology loves context. And causation cuts both ways. Are the methyl marks causing aging or riding along? It's both. Some marks are consequences. Some rewire gene expression enough to drive decline. Sorting, which is the work. Another guardrail. Reset too far and identity phrased. Cells that forget who they are don't help your heartbeat or your brain think. That's why partial reprogramming walks on eggshells. Drop CMIC to cool tumor drive, use tissue-specific switches, and build kill switches in case a cell goes rogue. The closer you get to rolling software backward, the more you brush against cancer's playbook. So where does this leave us? With a deeper clock that tells you how your life and your biology are talking, and a hint that some of that talk is editable. Not a master key, but a dashboard that finally shows the hidden dials. Slow the pace, and the rest of the system, telomeres, senescence, mitochondria, has fewer fires to fight. Reset carefully, and you might buy function without lighting fuses. It's software. It can be patched, but only if you respect the code. Why 120? Engineering limits in complex systems. So why does the line keep landing around 115 to 120? Not because one clock rules all, but because a lot of parts share tight tolerances. Think like an engineer. You don't design a bridge to last forever. You pick materials, stress limits, maintenance windows, and safety factors. Bodies are like that. Multicellular life runs on coordination, not raw power, and coordination sets ceilings. Start with the biggest headache, cancer policing versus regeneration. You need cells ready to divide, to heal cuts, replace gut lining, make blood. But every division risks errors, so the system installs breaks, shortening telomeres, senescence programs, apoptosis, immune patrols. Great for stopping clones, bad for endless repair. If you relax the brakes too much, precancers sprint. If you clamp down, tissues can't renew. That trade sets a lane. You can move within it, but drift too far either way, and you crash. Now zoom into parts with brutal specs. The heart beats about 3 billion times in a 100-year run. Valves don't get Saturdays off. Their leaflets slam and seal tens of millions of times a year in hot, salty fluid at pressure. Collagen crosslinks stiffen with age. Calcifications creep in. Tiny hinge points roughen. Surgeons can swap a valve, sure, but your native hardware wasn't built for 200 years of cycles. The tolerance stack-up hits you around the century mark. Kidneys filter your blood all day, every day. About 180 liters of fluid through tiny sieves called glomeruli. Each filter is a lacework of basement membrane and podocyte feet. Over decades, glycation gums the mesh, blood pressure scuffs it, immune hits scar it. You can lose half your filters and still be fine, because redundancy is baked in, but redundancy is not infinity. Past a point, your reserve runs thin and small bumps. Dehydration, a drug side effect, tip you into failure. Neurons are the stubborn ones. Most of your brain cells don't divide. They last as long as you do, holding memories, running circuits, maintaining miles of axons and synapses with proteins that need perfect turnover. Proteostasis drifts, misfolded proteins sneak into synapses. Mitochondria lag on long axonal highways. Microglia shift toward a hair-trigger inflammatory state. You can prune and rewire, but you can't swap the board. That sets a hard limit on how cleanly the system can run in year 130 without a catastrophic glitch. Meanwhile, the immune system pulls double duty as repair crew and riot police. Early in life, a hot inflammatory response saves you from infections. Later, that same tone cooks joints, vessels, and brain. Dial it down and you miss tumors and bugs. Dial it up and you shred tissues. Add thymus involution, fewer rookie T cells, and your adaptive playbook gets stale. You can vaccinate and boost, which helps a lot, but the base architecture still trends toward fewer clean options each decade. Stack in biomechanics we never think about. Bipedalism is great for hands and brain, but it loads hips, knees, and spine in ways four-legged cousins skip. Cartilage is avascular. It relies on slow diffusion of nutrients through motion. Once it thins and tears, chondrocytes can't sprint to fix it. You can regrow patches maybe, but the native maintenance loop wasn't spec-it for 150 years of upright wear, stairs, and weekend warrior basketball. Then childbirth. Our big brains and narrow pelvis made a deal. Human babies come out early, squishy-headed, and we carry a long dependency to finish wiring outside the womb. That's a win for cognition and culture, but it locks in pelvic mechanics and connective tissue tolerances that already ride a narrow line. Long life stretches collagen, shifts organ support, and complicates repairs that were never cheap. Even the wiring for staying warm and fueled has bounds. Endocrine networks, thyroid, insulin, IGF, adrenal stress axes balance each other. Tweak one to keep bones solid, and you overclock growth signals that cancer likes. Lower IGF-1 to slow aging in mice, and you stunt growth and fertility. Do it in people the wrong way, and you trade muscle for theory. Engineering again. Every gain shifts a load somewhere else. Here's a microbeat. Imagine a 115-year-old's day as a coordination drill. Heart valves a little stiff, kidneys running on fewer nephrons, bone marrow with tired stem cells, gut lining patching slowly, microglia edgy, cartilage thin. Each system still works, but with less slack. Then you get a flu. Fever pushes heart rate, dehydration taxes kidneys, inflammatory cytokines rattle the brain, appetite dips, muscle breaks down. Nothing exotic, just no margin. So why 120? Because that's roughly where the weakest links converge under current specs. Telomere budget meets senescence noise, meets immune drift, meets mechanical wear, meets neural single-use parts. Push one component past spec, say give telomerase broadly, and cancer policing fails. Fix cancer perfectly, dream big, and proteostasis still gums, neurons, valves still calcify, kidneys still silt. The ceiling isn't a single beam you can raise with one jack. It's the roof line set by many beams flexing together. Could better materials lift it? Maybe, piece by piece. Bioprosthetic valves that last longer, Senolytics to quiet noise, gene edits to tune repair, reprogramming pulses to refresh epigenetic software, smarter vaccines to cover immune drift. Those are beams strengthened. But unless you reinforce many at once, the roof line only inches. Engineering says you can extend service life by spreading loads and upping maintenance without breaking the system. Biology says the same. The 120 mark isn't a curse. It's the sum of trade-offs that let thinking apes survive long enough to teach their kids what actually moves the needle without fairy dust. Let's be real, most hacks are noise. The big wins are boring, repeatable, and they push a bunch of dials at once. Imagine grabbing 5 sliders and nudging each 10%. That's how you slow the pace your epigenetic clock ticks, cool chronic inflammation, and give repair a fighting chance. Start with sleep, not "I'll catch up this weekend." Real sleep, same window, most nights, 7 to 8 hours for most adults. Deep sleep is when growth hormone pulses, when the brain's glymphatic system rinses metabolic junk, and when immune memory firms up. Miss it, and IL-6 and CRP drift higher, insulin gets cranky, and your methylation clocks often edge older. Fix it, and you nudge a dozen pathways in your favor without swallowing a single pill. Next lever is muscle, not a beach body, force. 2 to 3 days a week of resistance work that actually challenges you. Muscle is glucose's best friend. It soaks up sugar after meals, flattens spikes, drops insulin demand, and tells mitochondria to multiply. It secretes myokines that tamp inflammation and wake stem cells in bone and brain. In older adults, strength training is basically youth insurance. Fewer falls, steadier gait, denser bones, better immune responses to vaccines. No joke, a barbell is more anti-aging than a most hyped supplement list. Protein quality matters because muscle doesn't build itself on vibes. Hit a sane target, roughly 1 to 1.2 grams per kilogram per day for many older folks, spread across meals, skewed toward lean sources. Pair it with resistance work and you tell the system, keep the scaffolding. Undershoot and you trade muscle for weight loss points that look good on a scale but read older on a frailty chart. Fiber is the quiet hero. 30 grams a day from plants, veggies, beans, whole grains, nuts, feeds a microbiome that makes short-chain fatty acids like butyrate. Those cool colon inflammation, tighten gut barriers, and even talk to immune cells. Your post-meal glucose swings shrink, your LDL creeps down, and your colon lining gets happier. It's also satiety without calorie math. Eat plants first, then whatever else. Vaccines are not sexy, but they're lifespan tech. Influenza, pneumococcus, shingles, Tdap, COVID boosters, each one cuts a hit your older self might not bounce back from. Fewer infections mean fewer inflammatory wildfires, fewer weeks of bed rest muscle loss, and fewer chances for latent nasties to wake up. It's blunt, and it works. Sunlight, carefully. 10 to 20 minutes on forearms and face in friendly seasons can keep vitamin D out of the basement, with mood and immune perks. If your skin or latitude says nope, supplement to solid levels. The point is tuned immunity, not tanning. Then glucose control, because roller coasters age you. You don't need perfect keto. You need smooth. Walk after meals. Front load protein and veg, push starch later. Sleep and muscle already did half the job. A continuous glucose monitor for a month can teach you which foods kick you. You don't need it forever. The result is calmer insulin, cooler inflammation, steadier energy. Social ties matter more than people want to admit. Loneliness spikes cortisol and pushes biology toward threat mode. Regular, real contact. Family dinners, a walking group, faith community, volunteering, tracks with longer health span in cohort after cohort. It shows up in clocks and in hospital charts. Boring, powerful, free. Sunrise and steps glue this together. Aim for 7 to 10K steps with some hills. Get morning light in your eyes to anchor circadian rhythms. Melatonin shows up on time. Sleep deepens. Toss in 2 short movement snacks on long work days. It's oil for stiff systems. Micro story from Blue Zone Life. In Sardinia, a shepherd in his 90s walks hills tending sheep, eats minestrone heavy on beans and greens, drinks a small glass of wine with friends, and sleeps when the village sleeps. He laughs often. He doesn't think about telomeres. He's pressing all the right levers. Muscle through terrain, fiber, modest calories, social rhythm, circadian alignment. He's not beating 120. He's making 95 feel like 75. What about supplements and drugs that actually help? A short list with caveats. Omega-3s can lower triglycerides and cool some inflammation if your diet's light on fish. Vitamin D if you're low. Creatine supports muscle, brain energy, and lifts training gains. Maybe a low-dose statin or blood pressure med if your risks point that way. Those 2 alone have banked more healthy years than most fancy molecules. Everything else sits behind sleep, muscle, fiber, and vaccines on the priority board. Habits beat hacks because they change context. A strong leg day flips on mTOR for growth in muscle, while AMPK rises between sessions to clean house, ebb and flow. A high-fiber dinner shifts microbiome output for hours. A set bedtime changes hormone timing for weeks. Stack them, and your methylation pace slows, your telomere attrition eases, and your senescence burden grows slower. You didn't hack one gene. You made the neighborhood calmer. Here's the honest fine print. Genetics load the dice. ApoE, FOXO3, and dozens of tiny variants tilt risk. Environment pulls the trigger. Air quality, walkability, job strain, access to care. You can't out-jog a hurricane or deadlift your way out of lead in the water. So advocate for cleaner air, safer streets, better food options. Your cells feel zoning laws. If you want a mini plan, lift twice a week, walk daily, eat 30 grams of fiber, hit protein targets, keep glucose steady, sleep on a schedule, get your shots, see friends. Then, maybe, layer a targeted therapy when trials say it's safe. Supplements, it's not flashy, biohacks, and wishful thinking. So after all that, the internet still says, don't worry, just take this pill and live to 150. Honestly, this is where people lose money, hope, and sometimes organs. Let's walk through the potholes so you don't drive into them at 80 miles an hour. Start with antioxidants. Sounds great. Quench free radicals, save cells. Plot twist. Your body uses tiny bursts of oxidants as signals to adapt. Exercise works partly because it stresses you just enough to trigger stronger defenses. Blanket that with high-dose antioxidant cocktails and you can blunt the training response. You feel productive. Your muscles get the memo, no need to improve. It's like bringing noise-canceling headphones to a fire drill, then wondering why the building didn't evacuate. Next up, NAD boosters. Nicotinamide riboside. Nicotinamide mononucleotide. The pitch, NAD falls with age. Top it up, power the mitochondria, save the day. In mice, bumping NAD can help certain tissues, especially in stress models. In humans, early trials move blood markers, but hard outcomes, strength, disease delay, are thin. Some forms are pricey urine with confidence. Meanwhile, push one pathway hard and others react. If a hidden tumor is sniffing around for fuel, a more energetic cell can be a mixed blessing. Could it help? Maybe, in the right person, the right dose, with eyes on the bigger network. Is it a universal youth serum? No. Metformin gets sainted every other Tuesday. It lowers liver glucose output, tweaks gut signals, cools insulin levels. In people with type 2 diabetes, it's a workhorse. In non-diabetics chasing longevity, the story is messy. Some studies hint at better mortality curves in diabetics on metformin versus non-diabetic controls, which sounds magical, until you remember confounders. In athletes, metformin can blunt training gains by dampening muscle adaptation. If your fasting glucose is fine, trading muscle for a theoretical nudge on a chart could be a bad swap. Tool, not talisman. Hormones are the siren song. Low T, low GH. Replace and feel 20 again, right? Here's the bill. Growth hormone makes things grow. Muscle, sure, but also organs, and maybe little precancerous patches you didn't know about. Chronic high IGF-1 tracks with some cancer risks. Testosterone can lift mood and muscle in deficient men, but unmanaged, it thickens blood, swells prostates, and alters lipids. In women, sloppy estrogen tinkering can raise clot and cancer risks without fixing the root. Sleep, muscle, stress. Hormones are powerful. Use them medically, carefully, with labs and clear goals. Not because an influencer said vitality. Peptides and gray market vials are the Wild West. BPC 157 heals everything. CJC ipamorelin boosts GH with no downside. Cool story. What's in the bottle? Who sterilized it? What's the real dose? Infections, allergic reactions, weird antibody responses. These pop up when sterile technique and quality control are more vibe than fact. Even legit peptides have trade-offs. Crank GH pulses, see glucose drift. Prod angiogenesis, watch a dormant lesion feel welcome. Clinics that promise no side effects are selling hope, not medicine. Hyperbaric oxygen, cryo, ice baths, saunas. Some of these have real effects in narrow lanes. Sauna use tracks with lower cardiovascular events in big cohorts, probably through heat shock proteins and vascular conditioning. Ice can help soreness. Hyperbaric has uses in wound care, and the bends, claiming it rewinds age wholesale, is a reach. Don't confuse a good stressor with a resurrection machine. Think seasoning, not new recipe. Fasting and keto wear halos. Short fasts and time-restricted eating can smooth glucose and trim calories without math. Great, but jump straight to multi-day fasts with meds or heart issues, and you can crash blood pressure, electrolytes, and judgment. Keto can help epilepsy and some metabolic cases. As a forever lifestyle, it can trade fiber diversity for ketones and push LDL into sketchy ranges in some people. Again, tool, not religion. Detoxes and cleanses? Your liver and kidneys handle that job 24/7. Most cleanses are laxatives with marketing. If you felt amazing, you probably just stopped eating junk for a week and slept more. Keep those habits. Skip the charcoal lemonade. The placebo effect is real, and I love it. Belief changes pain, energy, even immune tone. But placebo won't sterilize a vial or stop a clot. Feeling better isn't the same as being safer. Here's a micro story. A runner in his 50s stacks metformin, high-dose resveratrol, NMN, testosterone gel, and a peptide he bought online. He trains harder but plateaus. Labs show lower VO2 max gains than last season, fasting glucose oddly higher, hematocrit creeping up. He swaps half the stack for sleep, protein, structured intervals, and yes, boring vaccines. 3 months later, he's faster, his A1C drops, hematocrit normalizes. He didn't quit biohacking. He stopped fighting his own adaptations. So, the rule of thumb, if a claim skips trade-offs, it's selling you, not helping you. Ask, what's the mechanism? What's the human data? What's the downside? How would I monitor it? And what boring habit does this pretend to replace? If the answer is all of them, close the tab. Ethics and economics. Who gets more time? So let's say we nudge the ceiling, or at least make 95 feel like 75 for a lot more people. Who gets that? Because extra healthy years aren't just birthdays. They're money, work, care, and politics moving around. Start simple. Add 5 healthy years across a population, healthcare bends. Fewer strokes and fractures save billions. But pension math strains because people draw longer. Care work shifts too. More late life independence eases family burden, but the tail still arrives. Schools, jobs, housing, transit, everything skews older. And if wages and mobility don't flex, you get friction. Now widen the lens. Longevity tech rolls out like most tech. First to the rich, insured, and urban. If Senolytics, reprogramming patches, or bespoke cancer shields show up at private clinics at $30,000 a cycle, gaps widen. The top decile buys time and compounding advantage. More years to learn, invest, and pass wealth. The bottom decile waits, ages naturally, and pays the bill in work years lost. Health equity isn't a slogan here, it's the fulcrum. A society with 2 clocks, one for the affluent, one for everyone else, gets brittle fast. Work gets weird too. If 70 becomes mid-career, ladders jam unless we build ramps. Phase rolls, retraining credits, sabbaticals tied to skill refresh. Without that, the young stare at clogged top rungs and leave. Intergenerational trust phrase. Pair that with climate load. More people living longer in hot, stressed cities, and resource math tightens. Extra years must run cleaner and lighter, or we just stretch problems. Zoom into the lab, because access starts at design. Organoids, mini-organs grown from stem cells, help test drugs without risking people. But who owns the tissue? Did the donor get real consent or a checkbox on a clipboard? Embryo-adjacent work to study early senescence cues raises lines most folks haven't thought through. And germline edits, changes to eggs, sperm, embryos, don't just help one person, they echo down generations. There's no oops, roll it back after a generation is born. We need red lines before green lights. What's off limits? What's provisional? Who audits? Policy always lags science, so build guardrails now. 3 that matter. First, safety bars, independent registries, long follow-ups, kill switches in gene therapies, and liability that doesn't vanish in shell companies. Second, fair lanes. Public coverage for proven longevity care the moment it clears trials, not 10 years later. Pricing tied to value, with clawbacks if outcomes miss. Third, talent bridges. Paid retraining and age-open hiring standards so extra years don't trap people in yesterday's skills. Human moment. A nurse in her late 60s can still lift, think fast, and loves the work, but her hospital has a quiet bias for fresh energy. A modest senolytic course eased her knee pain. She wants 3 more good years bedside, then to teach. If the system says no because her birth year starts with 19, that's waste. The promise of more healthy time only lands if roles flex to receive it. Bottom line, longer health spans aren't a private hobby. They're public infrastructure. If we don't plan, the gains pool in the same places as everything else. If we do, 5 extra good years can pay forward in taxes, care, mentorship, and less suffering. Ethics here isn't abstract. It's who gets a seat, who pays the bill, and who writes the rules. When immortality turns on you, cancer as the anti-limit. So here's the price tag stamped in red on every live longer idea. Cancer. If aging is your body easing off the gas to keep order, cancer is the moment a cell cuts the brake lines and floors it. Push division to fix tissues, feed growth signals to feel young, turn telomeres back on to stretch the fuse. These are the same roads tumors use to go feral. Immortality, cell by cell, is their business model. Let's make that concrete. A normal cell keeps count with telomeres. Each division trims the tips. Get too short. Checkpoints flip P53 and P16, P21, and the cell either senesces or dies. It's a crude but effective dead man switch against runaway growth. Tumors survive by finding a crowbar. About 9 out of 10 human cancers reactivate telomerase, the enzyme that rebuilds telomeres. The rest turn to a back alley route called alternative lengthening of telomeres, where they recombine chromosome ends to fake a longer fuse. Either way, they break past the Hayflick limit and claim dividing rights indefinitely. What nobody knew at the time, back when telomerase felt like a fountain of youth, was just how often cancer would steal that fountain. Turn the enzyme on broadly, and you don't just help. Heart cells. You hand pre-malignant clones a credit card with no limit. That's why our cheat-the-clock experiments run inside cages of safeguards. Tissue-specific promoters, temporary pulses, kill switches if a growth marker spikes. Because the anti-limit isn't a theory, it's a bill collector with perfect timing. Meanwhile, look at the hallmarks of cancer as a mirror held up to aging. Aging leans on senescence to stop sketchy cells. Tumors resist cell death. Aging tightens nutrient sensing. Tumors rewire metabolism to guzzle glucose and glutamine. Aging's immune drift struggles to clear old cells. Tumors evolve cloaks, PD-L1 shields, Treg entourages to hide from what's left of surveillance. Where aging lets proteostasis slip, tumors scoop up chaperones to keep their hyperactive proteins from misfolding. 2 systems, same roads, opposite directions. Here's a lab bench picture that hits the gut. A pathologist lays out slides from an elderly patient who lived to 111. Arteries show calcifications, sure. Skin thins, sure. But the real surprise is the count of tiny, quiet neoplasms. Little clonal expansions in the esophagus and the thyroid and the bone marrow, field cancerization, that never made headlines because they stayed fenced in by senescence, short telomeres, and immune potshots. At 70, fences hold more often. At 110, the lumber's old. One storm, one inflammatory bout, one unlucky mutation, and a quiet patch goes loud. The ceiling feels like valves and kidneys, but the trap door is often a clone that finally got enough runway. Elephants and whales solve pieces of this, which is why they get to be huge and old without drowning in tumors. Elephants carry extra P53 copies, quick to trigger apoptosis if DNA smells off. Bowhead whales invest in better DNA repair and cold-tuned stress responses. Their CIRBP seems to help keep transcription tidy under stress. They also live slow, with long gaps between babies, in safer niches. Human life history sits in the middle, slowish but not glacier slow. We don't have their extra tumor suppressor copies or Arctic repair kit. Our workaround was to accept stronger limits on cell division and more senescence noise as the price of not dying of cancer at 30. Antagonistic Pleiotropy live on stage. Microstory. A mouse in a partial reprogramming study shows gorgeous muscle repair after OSK pulses. Under the microscope, fibers look younger, mitochondria brighter, epigenetic marks smoother. Then, in a different cohort with a slightly hotter dose, teratomas bloom in a few animals. Tissue blobs of cells that forgot their job descriptions. Same factors, same promise. One notch too high, and you've taught a handful of cells to ignore the rules. The team dials CMYC out, tightens timing, adds a suicide gene, gains shrink. Tumors vanish. That compromise, less youth juice, less risk, is the whole fight with cancer in one vignette. And there's a twist that makes this even harder. Senescence suppresses tumors early. Good, but a long-lived senescent cell's SASP can also stoke nearby precancers later. Bad. Clear senescent cells with Senolytics, and you may lower inflammatory wind that fans tumors. Good, but those sentinels also help wound healing and can flag danger. Bad if you clear them at the wrong time. It's a balancing act on a moving wire. Peto's paradox, the observation that big animals don't get proportionally more cancer, tells us the anti-limit isn't absolute. Evolution can stack anti-cancer traits if the environment buys time for selection to notice. But even those species didn't pick immortality. They picked better policing. Any human attempt to sail past 120 has to import that level of policing or invent its own. There's no version where we stretch the fuse and ignore border control. So let's be real about the road ahead. A path past the ceiling looks like layered defenses. Better DNA repair without sparking instability. Telomere maintenance, that's local, temporary, and paired with tumor sentries. Reprogramming pulses with hard-off switches. Immunotherapy-style surveillance tuned for aging tissues. And Senolytics scheduled so you keep the guards you need. Every step buys function, but touches the same levers cancer craves. If you don't engineer for that, you're not extending life. You're shifting because of death. That's the anti-limit in plain English. Immortality, cell by cell, is cancer. The way through isn't pretending that's not true. It's building systems that let cells act younger without forgetting where the stop signs live. In the end, our bodies choose order over forever. The near twin-twinning guardrail is how a thinking species stayed alive long enough to matter. Want the levers you can pull now and the moonshots worth watching? Subscribe, then jump to our deep dive on epigenetic clocks and the 3 trials that could move the line this year. Don't miss it. The story keeps unfolding.